Sorry, perfect.
Thank you. All right. Hello, everyone. I'd like to begin just in the spirit of reconciliation, acknowledging the Larrakia, the Dagaman, the Jawoyn and the Wardaman people as the traditional custodians of the land where we did this work and the land where I live. I pay my respects to their elders past and present, and I extend that respect to all Aboriginal and Torres Strait Islander people today. Today, I'm going to be talking about the role of honeybees in CGMMV epidemiology. And this work has been done by a team of people, Darsh Rathnayaka, Shreya Patel, Maxine Piggott, Brian Thistleton, and myself. So CGMMV, what is it? It's called cucumber green mottle mosaic virus. It's a plant pathogen within the genus Tobamovirus. It affects cucurbits, so watermelons, cucumbers, zucchinis, etc. And you can see some symptoms photos down the bottom of this slide. In the leaves, you see mottling and mosaic discoloration. You can get distorted fruit. And if you look in the far right-hand corner, there's a watermelon there, and you can see that it's got necrotic, rotten flesh inside. CGMMV is a virus that's mechanically transmitted. So if you touch a leaf and you break some trichomes on that leaf and then you touch another leaf, you can transmit the virus that way. It can also be moved in soil and via seeds. The virus was first discovered in England in 1935, and it's now distributed in more than 40 countries. And a lot of that spread in the distribution has happened in the last 20 years, primarily with the international trade of seed. It was first detected in Australia in the Northern Territory and Katherine in 2014, and it's since been detected in most states and territories. It's classified as endemic to the NT and WA, and it's under quarantine control in Queensland, New South Wales and South Australia. So why are we interested in this plant pathogen? Well, cucurbits are pollinator dependent, and in Australia, we primarily use Apis mellifera, the European honeybee, to pollinate cucurbits. And back when CGMMV was detected in Katherine in 2014, we started sampling in the beehives that were providing pollination services to those crops. And we found that the plant virus was present inside those hives. This was the first time this had been done anywhere in the world, and we managed to find CGMMV in all hive products, in the adult bees, the brood, pollen, the honey and the wax. More importantly, we actually looked at the viability of the virus that we were getting out of those hives. So that's looking at if the virus can cause infection. And we found that virus that we got from adult bees, pollen and honey was able to cause infection in healthy plants. So this identified a bit of a risk, and then we started thinking about questions that we needed to answer to enable us to manage that risk. I'm going to talk about some of them here, and if you stick around for the talk directly after me, Darsh will tell you the other half of the story. So the questions that I'm addressing are: how long is CGMMV detectable in beehives after initial exposure? Is the virus present in beehives viable, and for how long? Where is the virus coming from? Where are the bees picking it up from? And is it possible to reduce the viral load in hives using standard apiary management practices? So to look at how long CGMMV is detectable in beehives after initial exposure, we wanted to be sure that there was no reintroduction of virus into the hives. So we took some capped honey from some hives that we knew had been exposed to CGMMV positive plants. We cut portions out of those frames and we put them in a temperature controlled cabinet and stored them at 35 degrees, which is the standard temperature inside a beehive. We collected honey from those samples every month for 14 months. And then at the end of 14 months, we tested the 12, 13 and 14-month sample points using molecular methods. So obviously, the characteristics that I describe for what CGMMV looks like are pretty general. When you get discoloration and mottling on the leaves, that could be nutritional, it could be a range of different diseases or plant pathogens. When you're trying to find a plant pathogen on a bee, you've got even less chance of actually being able to see it. So we go through these range of molecular tests to find whether we have CGMMV on our bees. There's a range of conventional PCRs and a quantitative PCR. We visualize those results, purify and quantify them, send any samples we get away for sequencing, and then we put it all in a table and we analyze all those tests to then determine if we have CGMMV. This table here just represents the results from those 12, 13 and 14-month honey samples. So we've got the range of conventional PCRs, and we've also got the quantitative PCR, which generates a CT value. So for the CGMMV qPCR, we used 35 cycles, and we had our interpretation was that if you had a CT value less than 30, it was CGMMV positive. If you had a CT value in between 30 and 34, it was suspect. And if it was over 34, it was CGMMV negative. So in an ideal sample, you'd get a CT value less than 30, you'd get positive hits on all 3 conventional PCRs, and you'd get a sequence, and you'd be able to say that you had CGMMV. And essentially what I want you to get out of that table is that we were getting very strong hits for CGMMV present in that honey, and our CT values were very low. So that suggested that there was a high viral load remaining in the hive, or could remain in a hive up to 12 months after initial exposure. But the important question for us is, even if there is CGMMV in the hive, is it viable? And the only way to do that is to do plant inoculation studies. So we took some of that honey, we inoculated it onto indicator plants, and we used a range of cucurbits and native tobaccos. And then you grow those plants for 8 to 10 weeks in an insect-proof glasshouse. And at various stages throughout that 8 to 10 weeks, you test the plants for the presence of CGMMV. We tested the growing leaves near the inoculation point, roots, any fruit that were formed, and any seeds that came out of those fruit. And again, you put it all together in a table and you analyse it to say, okay, the virus is viable, it's capable of causing infection in healthy plants. And these are just some images of Shreya and Darsh setting up those viability trials in our insect-proof glasshouse. And the great result for us in terms of managing the virus is that we found that even though there was a high viral load found in honey 12 months after initial exposure, none of that virus was viable or capable of causing infection. Another question we had is, where is the CGMMV coming from? In previous work in the NT, researchers had identified a range of common weeds that were alternative hosts for the virus, and they grow around cucurbit production areas. So there was some question about whether the bees were picking up the virus from the commercial cucurbit production or whether they were picking it up from the weeds. So we had 100 pollen samples that we'd collected from hives over the past 7 years. And we sent those down to AgriBio in Victoria, where Joanne Mackey's doing her PhD. And she conducted metagenomic sequencing on those samples. In a subset of 54 of those samples, Joanne identified 366 different plant species from 72 families. And in each individual sample, we found a range of species from 1 to 38 species within a single pollen sample. So from this work, we got some pretty amazing data about host plant use by European honeybees in the Northern Territory. But unfortunately, we didn't get any clear correlation between CGMMV status and plant species identified. The final question that I'll be talking about is it possible to reduce the CGMMV load in beehives? So to do this, we got a positive beehive, a hive that had been exposed to CGMMV positive plants. And we just used some standard apiary techniques. We spun off the honey, we replaced the frames, we removed the top box, and we put that hive at a resting site, which is generally what happens after hives have been working cucurbits. We then sampled bees, brood, honey, pollen, and swabbed the surface of the wax from within that hive over a period of 14 months. This figure here shows our results. So on the x-axis, we've got time, and I'll just draw your attention to right at the end. We sampled up until 5 months, and then we came back 14 months later and we took another sample. So there's a bit of a time leap there. On the y-axis, these dots relate to CT values, which is that CT values from qPCR. If I can draw your attention firstly to the green dots and the orange dots, they represent adult bees and brood. And hopefully you can see that we don't get any detection of CGMMV in either adult bees or brood after 40 days post-exposure to CGMMV. We can detect CGMMV in other hive materials for a much longer period of time, but by the time we get out to 14 months, we can only find it in the honey. And obviously from the work that we've done previously, we've shown that that virus is not viable at 14 months. So this is promising to say that you can reduce the amount of CGMMV in your hive, particularly in the foraging bees. So in conclusion, CGMMV is detectable in beehives for over a year, but it's not viable. The pollen analysis is not suitable to identify the viral source where the bees are picking it up from, but the viral load within a beehive can be reduced by standard apiary practices. So this project was funded by the Mellon Levy through Hort Innovation Australia. We're winding up the project now, so we've got a range of fact sheets available on the Hort Innovation website with management recommendations and results if you're interested. I'd like to thank everyone who was involved, and particularly the growers and apiarists who supported this research and allowed us to conduct surveillance and collect samples from their properties. Thank you.
Thanks, Mary. There's a question in the chat there from David, who asks, he says, great and clear presentation, but also wondering how some of the hive samples did not have CCMV detected initially, then it was detected in later samples.
I think that's, David, that's just because one of the challenges with looking for a plant virus inside a beehive is that it's not actually moving around inside the beehive. So it's static. So it depends on which part of the beehive we sample. And that is one of the challenges with trying to find the plant virus inside the hive. You might take samples from 3 different pollen cells and only one of them contains CGMMV. So we tried to control for that by sampling from different places within the hive at different times and at the same time as well.
Have we got time for another question? If someone wants to raise their hand or put something in the chat. I guess I'll ask one, Mary. With your CT values that you got from your qPCR, you put the threshold at 30. Did you play around with that at all? And why did you pick that as the threshold?
I did not personally play around with that. That diagnostic technique had been established in a previous Hort Innovation project. And yes, it had been played around with a bit. And it was based on a range of samples that were collected over 5 years, both from plants and bees. I can put you in touch with the people who did develop that protocol if you'd like.
Oh, why not? It'd be nice. I wasn't sure if by wiggling it around it would change the results much. It's just, yeah, interesting.
Yeah.
Have we got any more questions before we move on? All right then. Well, thanks, Mary. If you want to stop sharing your screen, and we can move on then to Darsh